And also when we consider the classification of these animals, basically for the classification, and there are certain principles we consider, we call animal architecture and the fundamental characteristics. And as we said from the beginning, the level of organization of cells in the body of the animal is multicellular. Multicellular. Here is a single cell amoeba. Here is a multicellular animal. Different. So level of organization of cells in the body is multicellular in animals. And the body symmetry therefore vary. Some they do not have any symmetry. We cannot cut this animal into two because it is not equally dividing, asymmetry. Like Porifera. The first phylum we are going to learn, Porifera. They do not have symmetry. And some of the animals, when they are in advance, you can find they can be divided into several radial axis we call radial symmetry. For example, Cnidaria, phylum Cnidaria. And more importantly, when the animals advance more and more, they show bilateral symmetry. We call bilateral symmetry. We call them as bilateral because we can divide that particular individual only into two. Therefore, except the phylum Porifera, rest of all the animal phyla, they show any kind of symmetry, either radial symmetry or bilateral symmetry. Except phylum Porifera, rest of all the animal phyla, individuals show either radial symmetry or bilateral symmetry. Radial symmetry, and some of them show pentaradial symmetry. They can be divided only into 5 angles, like this way, 5 pentaradial symmetry, like Echinodermata, phylum Echinodermata, and the bilateral symmetry. And then the number of embryonic layers in the body. Again, some of the very early stage of animals, in the embryonic development, they do have only 2 embryonic layers, endoderm and the ectoderm. And they do not have a mesoderm in the middle layer, where middle layer is a cavity, non-living cell layer. Cavity filled with non-living cells or living cells. For example, Porifera. They don't have a middle layer. It is like a gel. But if you take phylum Platyhelminthes, you find the middle layer as a cavity filled with parenchyma cells. But when it is coming to the higher animals, you find middle layer during the embryonic development, it fills with dermal cells, mesodermal cells, where internal organs are going to develop in the fully formed individual. Therefore, depending on the number of layers of those animals, primitive animals, they have 2 body layers. They are diploblastic, regarded as diploblastic, and the advanced animals from phylum Platyhelminthes onwards, they have 3 tissue layers or cell layers in their embryonic development. They are regarded as triploblastic animals, diploblastic animals and triploblastic animals. Remember, diploblastic animals, they have 2 embryonic developmental body layers, example, Cnidaria. Triploblastic animals are the one from phylum Platyhelminthes onwards. Then, when it comes to more and more advanced type of animals, the coelomic development will take place. Coelomic development is the splitting of the middle layer, this layer, split over, forming a secondary cavity. That is called coelom. So coelom is the body cavity. And the body cavity or formation of the coelom usually therefore take place in between the digestive tract and the body wall, middle cavity. For example, Annelida, mollusca, Annelida, Echinodermata, and the arthropods and also chordates. Coelomic development, and some of the animals, for example, arthropods, coelom is reduced. Therefore, depending on the development of the coelom, we consider in the very early stage of animals, for example, platyhelminths. They are the ones who do have 3 cell layers of the most primitive type of animal category. 3 cell layers, most primitive type of animals, platyhelminths. But the middle layer do not have a cavity. So we call it as a acoelomate. Acoelomate means, A means negative, coelomate with addition letter A. Acoelomate, no internal body cavity. Then when we take nematoda and the Rotifera, 2 phyla, nematoda and the Rotifera, they are the body, you find the body, external body, wall, then the digestive tract. Middle is a liquid filled cavity. Liquid filled cavity, we call it as a pseudocoelom. Pseudo means untrue, false body cavity. So it is the advanced stage than the acoelomate, but primitive than the true coelomate animals beyond the phylum Nematoda, like Annelida, Echinodermata, Arthropoda, and the Chordata. Okay. So these categories of body cavity is acceptable only for triploblastic type of animals. That is why I said the most primitive triploblastic animal, that is platyhelminths. And Nematoda and Rotifera are advanced than platyhelminths because they have a pseudocoelom. Okay. That is how the coelomic development takes place in diagrammatically, and you can simply refer, and the formation of coelom. And there are 2 different ways the coelom is formed. One, it is applicable only for true coelomate animals like chordates, echinodermates, arthropods, and annelids. One is this coelom is produced by chrysozoa, like this, chrysocoelom. It is like, when the embryonic development, the embryo developed from the blastula here, and some of the cells move more interior into the cavity, interior to the blastocele site. And when they push towards interior, some of the cells form like separate in growth to form separate cavities, blastocele. But in other case, here, they form inward growth here, and from the formation of the gut, further development of inwards form separate cavities. 2 different ways. But you can see both appears ultimately similar to each. Here it is the diversion of the gut, that is enterocoelous. Here it is a separate type of cells forming separate cavities, we call chrysocoelous. So these are not necessarily to remember, that you have been learned during form and function, maybe during your A-level studies. And in these coelomic animals, some of the animals, if we take when the development of gastrula, where the blast pore pushes some of the cells most interior, and that particular opening we call blast pore here, blast pore. In some category of animal, this blast pore in the developed animal function as the mouth. We call it as protostomium animals, like arthropods and mollusks. But the blastopo in some of the animals, blastopo of some of the animals, animal categories, in the adult form, they develop as the anus. Instead of mouth, it is served as the anus. We call it as Deuterostome. And the Deuterostomians are the only two phyla, only one phylum, that is phylum Echinodermata. They show Deuterostomium and also phylum Chordata of the vertebrate. Rest of all the phyla are Protostomium. Protostomium means the mouth of the adult individual is the blastopo of the embryonic development. But Deuterostomes, that is the phylum Chordata and the phylum Echinodermata, anus is the embryonic development blastopo. Two different ways. This is how the cleavage pattern take place, radial cleavage, spiral cleavage, and also some of the animals, animal phyla, they have different larval stages. Different larval stages. For example, mollusk, phylum Mollusca, and the phylum Annelida. They develop through a larval stage called tropophore larva. This is the tropophore larva. And for example, phylum Echinodermata, when they develop as an adult, passing through a dipleurula larva, this is the dipleurula larva. Both are aquatic. Likewise, they have typical larval stages depending on the different phyla of the kingdom Animalia. And also they have different types of eggs. Some are indeterminate, some are determinate. And determinate eggs are usually the primitive type seen in arthropods, arthropods and lower animals. And the indeterminate eggs are usually seen in echinodermates and above. Determinate and indeterminate eggs means if we take an egg and the egg, if we take anterior, posterior, dorsal, ventral, and different cell areas decide which part of the adult animal is going to be basically based on the mother's genes provided into this egg. So these type of determinations are there in very primitive type of animals, individuals. But if we take deuterostomes, the eggs are not determinant. And the segmentation is also an advanced type of character. Usually segmentation is, you can see from phylum Annelida onwards. Annelida onwards, you can see the segmentation. It is an advanced type. Segmentation means each and every body part serve individually and function separately to each other. Segmentation. And also presence of notochord and vertebral column that decide whether the animals are vertebrate or invertebrate. If they have notochord and the vertebral column, they are vertebrates. And if they don't have, they are invertebrates. Likewise, we can divide individual animals into different categories. And the cephalization is also an important thing. As I said previously, cephalization is a developmental stage of bilateral symmetry. Bilateral symmetry. Cephalization improves the sensory organs to be located in anterior head area of the body. Head area of the body. And in the animal kingdom, if we take animal kingdom that you are going to learn from Monday onwards, there are nearly 35 known animal phyla. But we are not going to learn all these phyla. Some are we consider as important phyla, some we consider as not that much of import. Likewise, there are some phyla we can consider as very large phyla, very large, having so many huge number of species, large number of species. For example, phylum Arthropoda. And considering some of the other phyla, you can see there can be only very few amount of species. We consider them as minor phyla. But even if it is minor phyla, there may be important individuals. Therefore, even if some of the phyla become minor phyla, we have to learn those things selectively. So we have learned today how the scientific classification is considered, and because we have done the nomenclature on the previous day, and you have now learned the natural taxonomy hierarchy introduced by Linnaeus, and also within this time you came to know the phonetic approach and the cladistic approach in natural classification scheme, and the major characteristics used to classify animals in the animal kingdom. So that you have met all these objectives from the first day lecture as well. Method and importance of scientific taxonomy, why we have to classify and identify animals, and the natural taxonomic hierarchy, phonetic and cladistic approaches in natural classification, also major characteristics used to classify animals, and the importance of scientific taxonomy.